Hi, my name is Kristin Mussar, and I am conducting my research at the University of Washington in Seattle in the laboratory of Dr. Laura Krisa. Our laboratory has a long-standing emphasis on understanding mechanisms that can enhance islet tissue transplant survival and regeneration as a treatment for type 1 diabetes. Our goal is to discover what role these cell types play in the development of healthy insulin-producing cells and how they can be used therapeutically. Type 1 diabetes is caused by the body's immune system attacking its own insulin-producing cells. Initially, the body tries to regenerate these insulin-producing cells, but ultimately fails. We aim to discover ways to enhance the ability of insulin-producing cells to regenerate from both immature precursor cells and pre-existing insulin-producing cells. In order to study regeneration, we first look at how the pancreas naturally develops. This image is of the developing pancreas. If we zoom in to one immature islet cell, we can see that it has several decisions to make. It can either proliferate and produce more cells just like it, or it can mature into an insulin-producing cell. We have discovered that there are cells surrounding the immature islet cells that instruct the developing cells on which choices to make. These cells are called macrophages and are a type of white blood cell. Specifically, we discovered that macrophages that resemble those in the embryo instruct pancreatic progenitors to mature. When we looked at the developing pancreas, we found two different populations of macrophages surrounding the developing organ. These are the resident macrophages, shown in blue, and the inflammatory-like macrophages, shown in orange. Thus, our question is to determine which type of macrophages send the messages to the immature pancreatic cells that allow them to either A, expand in number, or B, mature into functional insulin-producing cells. An additional question we have is how are these cell types able to send the message from one cell to the other? We discovered that the numbers of each subtype is different in the embryonic and the newborn pancreas. Specifically, during early development, there are more resident macrophages, and at birth, there are more inflammatory-like macrophages. Again, resident macrophages are shown in blue, and inflammatory-like macrophages are shown in orange. Our goal is to discover what role these cell types are playing at each of the different stages and how they can be used therapeutically. We hypothesize that the role each macrophage type is playing is directly relevant to the developmental stage where we find the most of that subset. For example, during mid-gestation, the resident macrophages are more abundant. This is the point at which maturation of the insulin-producing cells occurs, and thus we hypothesize that the resident macrophages may signal the cells to not replicate while they are still learning to produce insulin. On the other hand, there are more inflammatory-like macrophages at birth. This is the stage at which the insulin-producing cells expand to form more insulin-producing cells. We hypothesize that the inflammatory-like macrophages are providing the signals that are telling the insulin-producing cells to undergo the process of expansion. In order to test my hypothesis, I will isolate embryonic pancreatic cells from donor mice and transplant them under the kidney capsule of recipient mice. In this host environment, either the resident or the inflammatory-like macrophages will be eliminated. The tissue will then be analyzed to determine how depletion of either macrophage cell type affects the development of the transplanted pancreas cells. We expect that elimination of resident macrophages will result in a relative abundance of inflammatory-like macrophages, and thus this will allow the insulin-producing cells to expand in number. On the other hand, when we remove the inflammatory-like macrophages, we expect that the insulin-producing cells will be unable to proliferate, and the size of the islets that are formed will be extremely small. If we now step back and look at the larger picture, the goal of this project is to find new treatments for diabetes. Macrophages can be used therapeutically in regenerative medicine in several different ways. They can be used to create more insulin-producing cells for transplantation. They can be used to keep the transplanted cells alive, and they may also be used to regenerate insulin-producing cells in diabetic pancreases. Our lab includes Dr. Laura Krisa, myself, Stephanie, and David. Additionally, we collaborate with the Cerulli Lab and the Institute for Stem Cell and Regenerative Medicine, also at the University of Washington. Thank you for your interest in our research.